Welcome to the Explainer. Today, we're going to decode the super high-speed messaging system that's running inside your body right now, the nerve impulse. It's that electrical whisper that lets you think, move, and feel all in the blink of an eye. You know, with the syllabus always changing, it's so easy to get lost in the details. But the core concepts, well, they're more important than ever. And honestly, it doesn't get more core than this. Understanding how a nerve impulse works is fundamental to, well, all of biology. So let's just dive right in. Okay, so first up, to really get how special this whole system is, we need to put it in context. Your body actually has two major communication networks, and believe me, they operate on completely different timelines. So on one hand, you've got the body's chemical system. Think of it like sending a message in a bottle. It uses hormones that float through the bloodstream, and eventually they get to lots of places and have these widespread, long-lasting effects. But on the other hand, you've got the neural system. This is a lightning-fast electrical grid. It's built for direct, immediate responses. It's literally the difference between snail mail and an instant message. All right, so let's zoom in a bit. Let's go from the huge network down to the single messenger that's responsible for all this lightning-fast communication, the neuron. Now, here's something you might not know. Neural tissue isn't just one type of cell. It's actually a whole team effort. You've got the star players, the neurons. They're the ones actually sending the signals. And then you have their absolutely essential support crew, the neuroglial cells. Okay, let's break this down. Take a look at the table here. The first big difference you'll see is excitability. Neurons are excitable. They're the only ones that can actually fire off a signal. The neuroglia? Nope, non-excitable. Their job is all about support and packaging. But here's the wild part. Look at the volume row. The neuroglia, the support crew, make up over half the volume of your neural tissue. So the support team doesn't just outnumber the stars, they take up way more space. And one of the most important jobs these support cells have is creating this fatty insulator called a myelin sheath. It wraps around the long part of the neuron, the axon. And this is just a perfect example of a huge idea in biology: structure dictates function. The way this sheath is built is exactly what lets the neuron achieve incredible speeds, as we're about to see. So before a neuron can even think about firing a message, it has to be in a constant state of readiness. You can pretty much just think of it like a fully charged battery, sitting there waiting for the signal to be used. But what does it actually mean for a cell to be charged and ready? I mean, it's not like there's a tiny lightning bolt inside, right? The answer is all about this delicate balance of electricity and chemistry. It all boils down to this number, -70 millivolts. A healthy, resting neuron holds this very precise electrical difference across its membrane, and we call this the resting membrane potential. This negative charge on the inside is so important. It's like a coiled spring, just holding a ton of potential energy, ready to be unleashed at a moment's notice. So how does it pull this off? How does it stay negative inside? Well, it's all about a carefully controlled imbalance of charged particles, which we call ions. The fluid outside the cell is just swimming in positive sodium ions. The inside has some positive potassium ions, sure, but the key is that it's also jam-packed with these big, negatively charged proteins that are just too big to leave. And these trapped proteins are what give the inside its overall negative vibe. So let's follow the flow here to see how the neuron keeps that negative charge. First, like we just said, you've got those big negative proteins trapped inside. That's factor one. Second, the membrane has these channels that are kind of leaky. And crucially, they leak more positive potassium out than they let positive sodium in. And finally, the most important player of them all, factor three, the sodium-potassium pump. This thing is the absolute workhorse of the nervous system. It's this tiny molecular machine that works 24/7. It uses the cell's energy money, ATP, to actively force 3 positive sodium ions out for every 2 positive potassium ions it lets in. So it's always losing that trade of positive charge. And this constant work is what maintains that perfect -70 millivolt potential, keeping the neuron's battery fully charged. All right, now for the main event. What happens when this charged battery finally gets the signal to go? A stimulus that's strong enough, we call it a threshold stimulus, hits the neuron. And you know what? It's just like tipping over the first domino. It doesn't matter if you flick it gently or push it hard. As long as you hit that tipping point, the whole chain is going to fall. The moment that threshold is hit, it is go time. These special sodium channels fly open, and suddenly that whole ocean of sodium from the outside comes flooding into the cell. This massive instant rush of positive charge completely flips the script, rocketing the membrane potential from negative all the way up to positive, and that's depolarization. But this positive state, it lasts for less than a millisecond. Almost instantly, those sodium gates slam shut, and a different set of doors, the potassium channels, swing wide open. Now, all the positive potassium ions that were inside rush out, taking their positive charge with them. And this mass exodus of positivity quickly brings the inside of the cell right back down to negative. And that is repolarization. And that's it. That's the whole show. This incredibly fast, all-or-nothing sequence of depolarization and repolarization. This lightning-fast spike in voltage from negative 70 millivolts up to positive 30 and right back down again. That is the action potential. That is the nerve impulse. It's the 1 or the 0 in your body's computer. But a message is pretty useless if it just stays in one place. So how does this little electrical spike actually travel down the entire length of the neuron, which can sometimes be over a meter long? Well, it depends on the type of neuron. In the unmyelinated ones, the ones without that fatty sheath, it's a slow, continuous wave, kind of like the tortoise. One spot triggers the next, which triggers the next. But in myelinated neurons, the ones that look like the rabbit here, something way more clever happens. Remember that amazing myelin sheath? It acts as an electrical insulator, which means the action potential can't happen along the coated parts of the axon. It can only happen in these tiny exposed gaps between the myelin bits, which we call the nodes of Ranvier. So the impulse is forced to literally jump from one node to the next. And this leapfrogging, it's called saltatory conduction, is what lets messages fly through your body at over 100 meters per second. It is ridiculously fast. So the electrical message has been created, and it's jumped down the axon at a breathtaking speed. But the journey isn't over. The impulse has now reached the very end of the line. The final critical question is this: how does this purely electrical signal cross the gap to talk to the next cell? Well, that's the story of the synapse, and it's the next step in this incredible journey.